A very good morning, ladies and gentlemen. I wish to take this opportunity to welcome you once again to our series of lectures. And today's lecture is going to be about the complement system. The complement system is very important because it is part of the natural immunity, the innate immunity. And it is very important because I know that if you lack some certain components of the complement system, you actually become very vulnerable to certain diseases. And so that's why it is very important for you to become as passionate about this topic as I am. My name is Stephen K, the resilient guy, and this lecture comes to you courtesy of the Immune System Explainer. So ladies and gentlemen, let's start by looking at the lecture outline. And we shall start with introduction into the complement system. Then we go to the pathways of the complement system, including the classical pathway, Mannan binding lectin pathway, the alternative pathway. And then we shall look at the effector functions of the complement system, and we shall look at regulatory mechanism of the complement system. And finally, an aspect I've not included here are disorders of the complement system. Introduction. The complement system is made up of about 30 proteins circulating in blood plasma. These proteins are encoded in the germline, and what that means is that they are inherited. You inherit them, and so that means you have some genes that encode them, and then you have them at the time of birth, and then they can manifest later within your blood plasma. The proteins circulate in inactive form. In biochemistry, enzymes that happen to be inactive are called zymogens. These proteins are activated by cleavage by a protease, which makes the next protein become a protease as well. And once activated, the complement proteins cleave and activate the next proteins. And this results in an amplifying effect, which is a cascade where one protein is cleaved, then becomes active, and then cleaves the next one, and then becomes active, and then cleaves the next one, and then that kind of cleavage continues like that to yield a very amplified effect of the cleavage. The complement system is activated right on the cell wall of the microorganism. That is where the membrane attack complex is going to be assembled, and the assembly of the membrane attack complex pokes pores or holes on the cell wall of the microorganism, weakening the integrity of the cell wall. And once weakened, the cell wall of the bacteria is going to allow contents of fluids or water to flow in, and then it's going to swell and may burst, losing its contents. Ladies and gentlemen, this is a very important image because I've brought it here specifically to show us where we're going to end after all the pathways of the complement system. We're going to end up generating what we call membrane attack complex. And the membrane attack complex, ladies and gentlemen, is like what you can see here. This is the cell wall of the bacteria. This is the internal environment, the cytoplasmic area of the bacteria. This is the exterior of the bacteria. And so as we talk about activation of the complement system, we're talking about assembly of this pore, of this MAC, membrane attack complex, right on the cell wall of the bacteria. And once assembled, it's going to be complete. This one is not complete, ladies and gentlemen. And once activated, as we talk about either contents of the bacteria or fluids flowing into the bacteria, we're talking about the use of this channel, which is the membrane attack complex, to either release things into the environment or have things from the environment get into the bacteria. that is what we call membrane attack complex. So now let's start talking about the activation, and we want to mention that we have three pathways through which the complement system is activated, and that is classical pathway, Mannan binding lectin pathway, and the alternative pathway. And ladies and gentlemen, this picture is very, very important because it's actually a summary of the entire complement system activation. And what happens here is that the three pathways of the complement system, starting from the classical pathway of the complement system, the Mannan binding lectin pathway, and the alternative pathway, all begin run differently up to this point. And this point that looks like a junction is a point at which we have assembly of the C3 convertase, which is an enzyme, the serine protease, that acts upon C3 as its ligand. So the three of them begin differently, and then they come and converge at this point of C3 convertase. From this point, ladies and gentlemen, the complement system usually progresses or proceeds as a common pathway, a single pathway, until this point, which is the ultimate point where the assembly of membrane attack complex is complete. Never mind about the rest of the things, because they're just mentions of some of the effector functions of the complement system, including the killing by MAC, opsonization by C3b, recruitment of inflammatory cells by C3a and C5a. We're going to be talking about that later. So let's start with the classical pathway. This pathway is initiated by an antigen antibody complex. So it is initiated by an immune complex, and that immune complex, the part of the antigen, could be a bacterial molecule that is bound to its specific antibodies and then forming antigen antibody complex. So the antibody, which is part of the immune complex above, will actually bind to C1q, and C1q is part of C1 molecule or complement system protein. C1 is a complex which is one of the complement system proteins circulating in blood plasma, and this complex is made up of 6 molecules of C1q, 2 molecules of C1r, and 2 molecules of C1s. So the binding of C1q activates C1s and C1r, and then activated C1s cleaves 2 serum proteins, and these 2 serum proteins are C4 and C2. The cleavage of C4, ladies and gentlemen, yields a large fragment called C4b and a small fragment called C4a. C4b binds covalently to the microorganisms cell wall, while C4a diffuses away, and then later it's going to be used in inflammation. So ladies and gentlemen, we've now begun to assemble MAC. We've begun to assemble membrane attack complex by having C4b attaching to the cell wall of the microorganism. After cleavage of C2, C2a is going to be added to C4b, again covalently on the cell wall of the bacteria, and together they're going to form C4b2a, which is the serine protease called C3 convertase of the classical pathway. The ligand for this enzyme, ladies and gentlemen, is C3, and I want to mention that C3 is the most abundant protein of all the complement system proteins. So let's talk about MBL pathway. The Mannan binding lectin pathway is very, very homologous to the classical pathway in many ways. It is initiated by MBL, which is a protein that is actually usually made in the liver. It's an acute phase protein, and this protein is very similar to C1q. MBL binds specifically to mannose sugars, that is on the cell wall of microorganisms. And MBL itself is actually a 6-headed molecule that makes a complex with MBL associated serine protease, that is MASP1 and MASP2. MASP1 and MASP2 are highly homologous to C1R and C1S of the classical pathway. That's why we say there is great homology between the Mannan binding lectin pathway and the classical pathway. Ladies and gentlemen, here we have a very, very important diagram again, or image, courtesy of Immunobiology, 6th edition by Garland Science. And this is the 6-headed protein that we just talked about. And what happens is that this protein is made up of MASP1, MASP2, and MASP is MBL associated serine protease 1, MBL associated serine protease 2, and that is how it looks like. That is how it looks like. Remember, we said this molecule binds to mannose sugars on the cell wall of microorganisms. Once activated, these proteases cleave C4 and C2, and C4, when cleaved, is going to give yield to a large fragment called C4b, and C4a, which is a small fragment. C4b, just like in the classical pathway, is going to be added to the cell wall of the bacteria in a covalent way, all right? And then C4a is going to diffuse away and become an inflammatory mediator. C2 is then cleaved and yields to C2A and C2B. C2A is added to the already bound C4B to make C4B2A, which is the MBL pathways C3 convertase. So ladies and gentlemen, up to that point, one thing that you're going to notice is that the C3 convertase of the classical pathway is the same as the C3 convertase of the Mannan binding lectin pathway. So the C3 convertase is similar to the classical pathways C3 convertase, and we've already seen that. Both of them are C4B2A, and this is the enzyme that acts on the ligand C3. Okay, then the last pathway is the alternative pathway. This pathway is initiated by spontaneous hydrolysis of C3. When C3 hydrolyzes spontaneously, it yields C3A, which is an inflammatory mediator, and C3B, which is bound to microorganisms in a covalent manner. Okay. So ladies and gentlemen, the assembly of C3 convertase of the alternative pathway includes another factor called factor B, which is also hydrolyzed to give rise to BA and BB. BB is then bound to C3B on the microorganism's cell wall, making C3B BB. This one here, okay, this one here is a C3 convertase of the alternative pathway of the complement system. C3B B, then capital B and lowercase b. This C3 convertase acts upon more and more C3 to produce C3A and C3B. C3B is added to the C3 convertase to form C5 convertase, which cleaves C5 to C5A and C5B. Okay, and C5B is very, very important because it's going to be the initiator of the common pathway that we mentioned earlier on, and then that is the thing that is going to drive the activation of the complement system until the end, until we have formation of MAC. Okay. Ladies and gentlemen, there is a very, very important diagram again here, courtesy of Immunobiology textbook, 6th edition by Garland Science of 2005. And ladies and gentlemen, this is now the ultimate thing in as far as the common pathway is concerned. As you can see here, now we have pieces of the C5b, C6, C7, being inserted into the cell wall of the pathogen. The pathogen is right here, the plasma membrane or the cell membrane of the bacteria is right here. And so we have C5b, 6, 7. So C5b, C6 and C7 complex bind to membrane via C7, and so they continue assembling on the cell wall of the bacteria. And then on the third column here we have C8 binding to the complex and then inserting into the cell membrane. You can see C8 inserting right here, and we have C8 molecules bind to the complex and then polymerize. So you can see what is happening here, and the assembly of MAC is still progressing here. And finally we have 1 to 16 molecules of C9 bind to form a pore, and so the pore that I illustrated before is now complete and has been inserted into the cell wall of the microorganism. Now, ladies and gentlemen, that is how the complement system is activated. There are several effects that come as a result of the complement system activation, and these are the things that we're calling effector functions of the complement system. And these include cell lysis. This is going to be done by MAC. We've already demonstrated how MAC is going to be inserted into the cell wall of the microorganism, and so the insertion of MAC is going to poke some holes, some pores throughout the cell membrane of the bacteria, causing loss of integrity. The other, so what happens here before I can navigate away is actually the bacteria now losing the contents or lysing as a result of attracting fluids and water from the environment and then bursting. That's what we're calling cell lysis. Then we have opsonization. C3b is an opsonin, ladies and gentlemen. It has the ability to coat the foreign antigen. The bacteria or the fungi in that case is going to be coated, and then because phagocytic cells have receptors for C3b, then what that means is that once bound by the C3b, bacteria are going to be more vulnerable to phagocytosis by phagocytic cells like macrophages, dendritic cells, and neutrophils perhaps. Okay. Then the third effector function of the complement system is chemotaxis. C5a, C3a, you'll remember we said they were to diffuse away to go to inflammation, and so they're going to participate in inflammation by attracting phagocytic cells to the site of injury, and then they can phagocytose the microorganisms at the site of injury or at the wound. And then the other very important aspect of the effector function of the complement system is immune complex removal. We said that the classical pathway of the complement system is initiated by antigen antibody complexes. So the utilization of these complexes is going to reduce complexes within the body, and that is going to help in preventing certain disorders that are caused by immune complexes, and these for the most part are actually autoimmune diseases. The other effector function of the complement system is C3d generation. The degradation of C3b generates C3d, which attaches to the cells with the complement, with the help of the complement receptor 2, and that is CR2. This, ladies and gentlemen, enhances immune responses. The cells with CR2 include the B cells and the dendritic cells, and sometimes the T cells. Okay. The last part of this lecture, ladies and gentlemen, is regulation of the complement system. Regulation of the complement system. Although we are also going to talk about a few disorders of the complement system. Now, to prevent autoimmunity, okay, and autoimmunity is actually a situation whereby the immune system attacks host cells or your own cells. The complement system must be regulated, and there are some proteins that are actually also encoded for in the germline that happens to be within your blood plasma that do this regulation. The cleavage of C3b to the inactive form called IC3b, formed by factor I, is very important because it averts the formation of C3 convertase. Of course, we've known the value of C3 convertase because it acts and hydrolyzes C3, which is the most potent, the most abundant protein of the complement system. And so the inactivation of it means no assembly of C3 convertase of the classical pathway, of the non-binding lectin pathway, and of the alternative pathway. The other point of regulation is the complement receptor 1 and the decay accelerating factor DAF. These two compete for with factor B for the binding place on C3b on the cell surface. So that means if these two proteins, CR1 and DAF, are going to outcompete factor B, then that means they're going to displace B, and what this in essence means, ladies and gentlemen, is that we're not going to have the C3 convertase of the alternative pathway, and that slows the process of activation of the complement system on the cells of the host. The other point of regulation, ladies and gentlemen, is C1 inhibitor, C1INH, which binds to C1R and C1S components of the classical pathway, and this shuts down the proteolytic activity. This stops the activation of C4 and C2, and that as well means we're not going to have assembly of the C3 convertase of the classical pathway. Now, ladies and gentlemen, let's briefly mention something about the disorders of the complement system. What would happen if you did not have the 30 proteins that we talked about? If you have defective genes that encode those 30 proteins, or some of them, what would happen to you? That is a question we want to answer. Now, a deficiency of C3, which is genetical, has been found by scientists to predispose individuals to frequent events of bacterial infections. A deficiency of C2, as well as C1q, C1r, and C1s, or C4, has been found to cause immune complex disorders. Another disorder of the complement system, ladies and gentlemen, is a deficiency of C1 inhibitor. A deficiency of this regulatory protein produces hereditary angioedema, and the massive release of anaphylatoxins C3A, C5A may cause edema of the airway and the skin. And that is it for today, ladies and gentlemen, and we are very, very grateful for your time to have followed what we're talking about. And if you liked this lecture, please subscribe to help us take the world and to be able to get updates from our YouTube channel every time we publish a video. We also appeal to you to subscribe to our main website, immunostudies.com, and when you become a member of that site, it's going to be very good. Thank you very much, ladies and gentlemen.